Welcome to Module 4. So in this module, we're going to cover three major topics: respiration, photosynthesis, and then signaling and receptors. These are our learning objectives for part 1. So respiration, aerobic respiration, is a complete oxidation of glucose to carbon dioxide and water in the presence of oxygen. It is how we use our food to make the energy that the cell needs in the form of ATP. Respiration is going to involve several steps, and this is the overall formula you're going to need to know for aerobic respiration. Keep in mind, respiration can also occur without oxygen, anaerobic respiration, but this is not going to create nearly as much ATP. So an overview of respiration, it's going to involve 5 major steps: glycolysis, the preparatory step, the citric acid cycle, also known as the Krebs cycle, the electron transport chain, and then ATP synthesis. This video will cover an overview of these steps. Remember, you will need to know these processes in more detail. See the textbook. So glycolysis, we're going to make some ATP here. We're going to start with a 6-carbon glucose, and we're going to end with 2 3-carbon pyruvate molecules. And from this, we're going to gain 2 ATP and 2 electron carriers called NADH. This first step does not require oxygen, and it occurs in the cytosol. And glycolysis is broken into 3 major phases. The first phase uses the addition of 2 phosphate groups to split the molecule into 2 3-carbon molecules. In phase 2, the cell is going to make some ATP and NADH, and in the final step, we're going to create the last of the ATP and end with 2 molecules of pyruvate. These are the overview of the steps of glycolysis, again, in those 3 phases. If there is no oxygen, the cell can undergo fermentation to process the pyruvate into either lactate or ethanol and carbon dioxide. If, however, there is oxygen, then we can proceed with aerobic respiration. So again, we started with glycolysis. The remainder of these steps are going to occur in the mitochondria. And next we have the preparatory step. So this is showing you that pyruvate will be transported into the mitochondria. It'll undergo a preparatory step called pyruvate oxidation, the citric acid cycle, the electron transport, and then eventually ATP synthase. So pyruvate is first oxidized to acetyl-CoA, and we're going to start with those 2 3-carbon molecules of pyruvate, and in this step we're going to lose 1 carbon from each of them as carbon dioxide, and we're going to give electrons and 1 proton to NADH, gaining NADH. So then acetyl-CoA can then enter the citric acid cycle. It will take 2 rounds to process the 2 2-carbon molecules of acetyl-CoA. And this 2-carbon CoA group is going to be added to oxalo acetate, creating citrate. And then as the cycle progresses, we're going to lose those 2 carbons as carbon dioxide. And from this, the cell will make ATP as well as NADH and another electron carrier, FADH2. This cycle is shown for just 1 molecule of CoA, and so it will take 2 rounds to process both acetyl-CoA molecules. This is the cycle shown in full, again, tracking those carbons that come in, and then we have carbons that come out. Now that we have done the citric acid cycle, we can then proceed to the electron transport chain, which again also occurs in the mitochondria. We have all of these reduced enzyme-carrying coenzymes, as NADH, the FADH2, and we need to use them now to make ATP. The electron transport system is going to shuttle the electrons through protein complexes with cytochromes oxidizing the electron carriers until we get to oxygen, which is our final electron acceptor. At the same time, these complexes are also going to pump protons, creating a gradient. And this is showing you that the electrons are going to move down their energy, starting either from the NADH or FADH2 enters here into complex 2. All of them will then pass into complex 3 and 4 before finally being shuttled to oxygen by complex 4. And at the same time as the electrons are moving, we're going to be pumping protons, creating those gradients. Now, the proton gradient is going to be used by an enzyme called ATP synthase, which has 2 components. It's got an FO component that is going to drive the proton translocation, and the F1 component, which actually makes the ATP. So in this video, we covered uh, respiration and how aerobic respiration uses glucose and oxygen to make ATP. In the next video, we're going to cover the steps of photosynthesis.